Hello, my name is Monica, and I will be discussing an article by Korea and her research team that designed a gamma SARS-CoV variant adapted subunit vaccine that was known to induce broadly neutralizing antibodies against SARS-CoV-2 variants and protects mice from infection. Here I will briefly outline the study's hypothesis, methodology, and main findings related to the research objectives. As you may know, severe acute respiratory syndrome coronavirus, also called SARS-CoV-2, is a virus that is known to have caused the COVID-19 global pandemic that infected over 777 million people worldwide and resulted in 7 million deaths. Several variants of concerns (VOC) continues to emerge, including the Beta, Delta, Gamma, Omicron, and other subvariants. The vaccines authorized for emergency use or fully approved are safe and highly effective against severe disease. However, it has been reported that vaccine-induced protection against symptomatic SARS-CoV-2 infection reduces over time, and this feature creates the need for new vaccine strategies that can induce more potent, durable, and broader immune responses to enhance protection against SARS-CoV-2. The first approved vaccines were mainly directed against the spike protein of the prototype Wuhan 1 SARS-CoV-2 strain. Therefore, their efficacy against certain VOCs is limited. Thus, vaccines are being adapted to variants as a strategy to improve vaccine effectiveness. Most of the variant-adapted vaccines developed were bivalent or monovalent, containing beta and ancestral spike protein as antigens. Monovalent and bivalent vaccines refer to different types of vaccines based on the number of antigens they contain. A monovalent vaccine targets a single antigen or strain of a pathogen, and it targets providing immunity against a specific disease caused by that particular antigen or strain, whereas a bivalent vaccine contains antigens from two strains, for example, alpha and beta strains of COVID. It provides immunity against two specific diseases. The figure on the right displays the structure of the virus particle comprising of the spike protein, which contains the receptor binding domain, RBD, and its two subunits, a common target used as an antigen in the making of a COVID vaccine. The gamma variant contained mutation in its RBD, the spike protein, and thus the authors designed an antigen RNA accordingly to create the gamma-adapted vaccines. The authors of this study developed a gamma-adapted RBD subunit vaccine adjuvanted with aluminum hydroxide to assess its immune responses against different VOCs and compare its efficacy rate against the ancestral prototype vaccine. Additionally, they performed clinical studies using the designed vaccine to observe neutralizing antibodies and antigen-specific cellular immune responses in mice. To begin the study experiment, they first constructed the SARS-CoV-2 ancestral and gamma variants antigen, which consisted of the peptide sequence of the SARS-CoV-2 spike protein. Both constructs were codon optimized with respect to the mutations in the RBD region of both variants. Then they were cloned into PJC3UMC2 expression vector. These recombinant plasmids were linearized and were transfected into Chinese hamster ovary cell line culture by liposuction, which uses a lipid complex to deliver DNA to cells. The plasmid clones were selected using ELISA technique that allows for specific antibody-antigen complex to be isolated. A-week-old female BAL-BC or C57BL6 mice were used to examine the immunologically related responses elicited from both the ancestral and gamma-adapted RBD vaccines. The mice were intramuscularly inoculated at day 0 and day 14 with gamma RBD adjuvanted with alum, ancestral RBD alum, or a placebo containing alum alone in separate mice groups. Blood samples were collected weekly to measure total and neutralizing antibody titers, and at day 42 post-prime immunization, animals were sacrificed and their spleens were harvested. RBD-specific antibody responses, specifically IgG, from mice subjects were evaluated by indirect ELISA. This technique involves coating plates with either gamma RBD or ancestral RBD antigens in PBS for incubation. The plates are then washed and incubated with diluted sera and are washed and incubated with the polyclonal HRP conjugated anti-mouse IgG to target IgG proteins in the sera. The samples are then subjected for optical density readings at 450 nanometers to determine and assess the IgG levels elicited from both types of RBD antigens. Lastly, a SARS-CoV-2 neutralization assay was conducted to examine the effectiveness of vaccine in neutralizing the SARS-CoV-2 virus. A neutralization assay is a laboratory test used to evaluate the ability of antigens to neutralize the ineffectivity of a pathogen. To perform the SARS-CoV-2 neutralization assay, serum samples obtained after vaccinations from mice were used to incubate with the SARS-CoV-2 virus, and the cytopathic effect (CPE) of the virus on the viral cell monolayers, which are cells derived from normal kidney cells, are assessed by surface scanning at 585 nanometers in a microplate reader, where the average optical density of each experimental sample was used to calculate the percentage of neutralization. Immunogenicity of vaccine formulations was assessed in BC mice after intramuscular immunization in a 2-dose vaccine schedule, using each constructed variant antigens adsorbed to aluminum hydroxide. The RBD of gamma and ancestral-specific IgG was evaluated by ELISA technique in sera of immunized animals. According to Figure B, anti-RBD IgG elicited from gamma-adaptive vaccine produced slightly more IgG titers, whereas the IgG titers that are stimulated from anti-ancestral RBD were observed to be slightly lower in concentration over the course of 40 days. On the other hand, anti-RBD IgG titers from ancestral RBD alum vaccine produce similar levels between the anti-gamma RBD and anti-ancestral RBD. However, after 21st day, the IgG titers levels drop slightly and increase slightly after the 35th day, while the anti-gamma RBD was observed to gradually increase at a slow rate and sharing a plateau at 40th day with the anti-ancestral RBD. Overall, both vaccine formulations induced high levels of IgG antibodies against the ancestral and gamma RBD antigens. In addition to determining the IgG levels, the neutralizing antibody titers in sera were also determined for each group of vaccinated mice at different time points, evaluated by a live virus assay using ancestral or gamma SARS-CoV variants, which is depicted in the figure on the left. The titers correspond to the 50% of virus neutralization, NT50. The gamma RBD alum immunization with the formulation containing gamma RBD plus alum elicited high neutralizing antibody titers against both variants at all times of immunization. In contrast, ancestral RBD antigen induced higher neutralization titers against ancestral SARS-CoV-2 than against gamma variant until day 35 post-prime immunization. The figure on the right displays the neutralization antibody titers against ancestral SARS-CoV-2 and different variants evaluated at day 42 in both immunized groups. It shows that gamma RBD plus alum vaccinated mice induce a long-lasting antibody response with a broader neutralization activity. Vaccine development procedures heavily rely on biotechnology at various stages of the process, from initial research to production. Biomolecular techniques that are often employed in the biotechnological field, such as genomics or proteomics, are used to identify antigens that can stimulate an immune response against specific pathogens. This involves sequencing the genome of a pathogen to identify potential target proteins that could serve as antigens for vaccine development. Additionally, the recombinant DNA technology also allows for vaccine design using vector systems to introduce foreign DNA for cloning that can be purified for further use as mRNA technology. These advanced techniques enable the production of vaccines that can mimic the structure of pathogens or deliver genetic material to produce antigenic proteins, triggering an immune response. The authors preceded vaccination with gamma RBD plus alum on K18HACE2 transgenic mice to examine the overall protection from infections rising from only one variant type, which was the Omicron SARS-CoV-2. Testing vaccinated mice against other variants might have shown a broader comparison in how well the vaccine protects the infected animal from the virus. Other than this, the study did an exceptional job at testing various research objectives. And this comes to the end of my presentation. Thank you for listening.